Good morning. Good morning, everybody. Welcome to Medical Grand Rounds and the John Butler Endowed Lecture. My name is Bill Altemeyer. I'm the director of the Center for Lung Biology, and it's my privilege today to introduce our guest lecturer. But before I do that, I just want to tell you a little bit about this lecture and John Butler. So Dr. John Butler was recruited to the University of Washington in 1964 to build the I can see a spirit is here. To build a new division of respiratory medicine. Dr. Butler's leadership is what the Division of Pulmonary and Critical Care and Sleep Medicine has flourished from, and with its focus on clinical excellence, research, and education. Past John Butler lectures have often also included personal anecdotes about his character and interests. Fortunately, my time here at the University of Washington did not overlap his, so I'm not going to share any of those today. But I am grateful for the legacy that he gave the Division of Pulmonary and Critical Care Medicine, its focus on research and education and clinical excellence, because I've benefited from that, as has our lecturer for today. So now I want to introduce you to Dr. Janet Lee. Janet embodies the same academic ideals that John Butler did. Janet received her medical degree from Georgetown University and then did her residency in internal medicine at University of Alabama in Birmingham. Following that, Janet moved to Seattle at the University of Washington for Pulmonary and Critical Care Fellowship, and we overlapped during her time here. After her clinical training, she entered a research fellowship under the mentorship of Dr. Tom Martin, and subsequently was recruited to the faculty at the University of Pittsburgh, where she has remained since and flourished. She is currently a professor in the Department of Medicine and in the Department of Environmental and Occupational Medicine. She maintains her own lab and is a world-renowned expert on host defense and host interactions with pathogens. And in addition to that, she's the director of the Biorepository and Clinical Translational Core for the Division of Pulmonary and Critical Care Medicine. In addition to her clinical and research interests, Janet has a passion for education and training the next generation of research scientists. Janet has a lot of students, residents, and fellows always rotating through her lab. She's the director of the Masters of Science Translational Medicine Core at their division. When I see Janet at conferences, she always has 3 or 4 trainees with her who she's aggressively introducing to everybody she meets and strongly advocating for attendance at their presentations. So I think that when we look at Janet's career, her focus on research, her passion for education, she really embodies everything that Dr. Butler stood for and is a great speaker for us to have here today. So please join me in welcoming Dr. Janet Lee.
Thank you so much for that kind introduction. I'm going to put this down and then start this one. Okay. I just wanted to thank Dr. Glennie and Dr. Bill Altmeyer and the rest of the faculty within the Division of Pulmonary for inviting me to come out here to Seattle to give this talk. I'm deeply honored to give the John Butler Lecture for 2019. Many years ago, I was a fellow here, and I did not know or have the personal connection with Dr. Butler before his untimely death. But upon reflection, as I was focusing on this lecture, I realized that I am the direct recipient, like Bill said, of the legacy that he created. I think here, it was here that I learned my passion for science and pursuit of discovery. And here I was enriched by the true community of colleagues and learned the value of mentoring and teaching the next generation. So it is only with gratitude that I give this lecture titled Host Defense and ARDS, Two Sides of the Coin. I have no conflicts of interest with any specific commercial entities. And I'll begin with a clinical objective, and that's to increase understanding of the definition of ARDS and kind of bring everybody on the same page and really focusing on the risk factors for the development of ARDS. And then I'm going to pivot a little bit in the second objective and discuss a story that we've been developing in the laboratory. And really it's predicated on the idea that imbalance in host defense and immune regulation as an underlying driver of ARDS. So Dr. Sachs is a master clinician at one of the hospitals at UPMC. And this was actually an interesting quote that appeared in a local newspaper. He was trying to describe the structure and function of the lungs to the lay community and his patients. And how he described it was a branching tree, and there was millions of air sacs, and he described them as grapes. And clearly that's what it looks like here in this cartoon. But I want to and he also talked about the function, which primary function, as you know, is gas exchange. But I want to direct you to this electron micrograph, which is the alveolar epithelial endothelial capillary membrane. And this is really a very thin membrane. It's approximately less than 5 microns in width. And if you compare it to human hair, which has the thickness of approximately 75 microns, you can see really how thin it is. But fundamentally, this is where disruption can occur in acute lung injury. And a diffuse disruption can cause extravasation of proteinaceous fluid into the air spaces and cause impairment in gas exchange. So what is ARDS? Well, the initial description, as many of you know, a lot of my pulmonary colleagues, originated from an article in Lancet in 1967, and the first author was Dr. Asbaugh. But it was a case series of 12 patients, 7 of whom went to autopsy. But what they described was severe tachypnea, hypoxemia with respiratory failure, and diffuse airspace opacities in the lungs. And they exhibited impaired compliance or stiff lungs, and they were very responsive to PEEP. But on autopsy, what they showed was it bore similarities to respiratory distress syndrome in newborns with formation of hyaline membranes, which essentially are cellular debris and proteinaceous debris that line the alveolar ducts and air spaces. And so the idea of ARDS, that something that was identified in newborns could actually happen in adults, was conceived. So why was it that we didn't know about ARDS before that time? Well, we did, but it really couldn't have happened without the advent of modern critical care medicine, i.e. mechanical ventilation, because these were patients in extremes of respiratory distress. And so this occurred in the 20th century, and as you can see, mechanical ventilation, we were able to store safely and depot blood transfusions. We developed the dialysis machine, and then war was what catalyzed all of this. And so World War I and World War II, there were reports of healthy soldiers that succumbed to catastrophic traumatic injuries. And they also showed wet lung. And then in Vietnam, it was called shock lung or Da Nang lung. The definition of ARDS has also evolved over time. And now we're in the current era, 2012, what we call the Berlin Consensus definition. And what it showed was 4 criteria: acute in onset, occurring within 7 days. So set what acute means. And then, of course, it would have to have diffuse pulmonary opacities consistent with pulmonary edema. But then they included risk factors or the origin of edema. So we had to know that hydrostatic edema or volume overload, heart failure, was not the major cause of this edema. And then obviously the hypoxemia severity was based upon the PaO2 to FiO2 ratio. And these are the risk factors that they delineate in the Berlin definition. This is direct injury, where direct against the lungs. So the most common is pneumonia, but you can see a list of different risk factors. And then indirect, with the most common being non-pulmonary or extrapulmonary sepsis. So The pros of this ARDS definition is that, well, it provided more explicit criteria for radiographic abnormality. So that was very useful. But, and also risk factors and the origin of edema. However, there are some cons associated with it. It still traditionally requires an invasive arterial blood gas. And so in low resource settings, that's not always possible. So does this person have ARDS or not, or can they be included in a clinical trial or not? So I think these are things that people are trying to sort out and clearly addressing. And then the interpretation of the radiographs is still a problem. And I think adjudicating whether or not somebody has ARDS still, I think, is a challenge for even experts. And because it's clinically based, there's some imprecision regarding classification. But I think overall, this definition has provided a way to move forward with clinical trials, with really studying the disease across the world. So this is one clear example of some of the issues related to the clinical definition, as many of my colleagues know. So which chest x-ray is consistent with ARDS? And this is an online supplement to the Berlin definition. And I can just tell you that it's these two on the left that would be consistent with bilateral pulmonary airspace opacities here. And then these look more like consolidations, but they dominate both lung fields. So using this current definition, people have looked at the epidemiology of ARDS in, globally. And so this was a lung-safe study, as many of you know, and three take-home points, at least the way I see it, was that using this current definition, 10% of patients in the ICUs will globally have ARDS. And then, as expected, mortality is associated with the severity of hypoxemia in the ARDS patients. And then what was really interesting was that the risk factor, 59% had pneumonia and 15% had extrapulmonary sepsis. So in other words, if you were going to study and model ARDS in the laboratory, I think you could pick infection as a model system. This is also another caveat to the ARDS issue, and it's looking at globally the infection in ICUs. If you look at any one given day, and this is a cross-sectional analysis, 51% of patients in the ICU are considered infected, with 71% receiving antibiotics. And not surprisingly, the most common site of infection are the lungs. And 70% had positive microbiology data, with 62% showing gram-negative organisms. And so what this tells us is that infections are very common in the ICU. And ARDS patients, it's exceedingly difficult to know, even if their risk factor was not infection, but they're in the ICU, whether or not they're infected or not, particularly because many of them had abnormal chest x-rays to begin with. So it's incumbent upon the clinicians to really scout this out during their stay. This is looking at sort of the association with outcomes in infection in the ICU, and not surprisingly, high rates of infection, longer ICU stays, and then if you are suspected to be infected or have an infection, it doubles your ICU mortality rate as well as your hospital mortality rate. So infection is a major issue in our ICUs. And so when we were trying to model an important question in ARDS, we obviously focused on host defense. And so I was going to take the point analogy and show on one side, we need a vigorous host defense to fend off pathogens. That's very clear. We have that we know. But on the other hand, the host needs to regulate unwarranted inflammation to prevent tissue damage. And so our interests stem from trying to identify host determinants that regulate the inflammatory response. So one question that we were really interested in was, what are the molecular determinants of host resilience in lung injury? And there were three things that we wanted to look at. One was maintenance of the barrier function, preventing or reducing pathogen-initiated host-sustained tissue damage, and then promoting repair and remodeling. And I'll go on to say what resilience means. In host microbial interactions, we talk about host tolerance, host resistance, and host resilience. And resistance is the ability to clear the pathogen. Tolerance is to actually tolerate the pathogen without any perceived pathology or observable pathology. And then resilience is really to recover from the pathogen. And we thought that what was the most important outcome is host resilience. However, in really trying to model the disease process, those three things are not mutually exclusive, and they're closely intertwined, is actually the take-home message. So we were interested in a matrixellular protein called thrombosponin 1. And this protein is secreted by a number of cells during injury. And thrombosponin 1 concentrations have been shown by Adele and colleagues many years ago to be increased in the air spaces of ARDS patients. And the concentrations correlated with composite injury scores. One of the cells that secrete thrombosponin 1 are platelets. And so it's on a per mass basis, major constituent of alpha granules. And when platelets are activated, they secrete thrombospondin as well as activated endothelial cells during injury, and perhaps a variety of other cells such as stroma cells in the lungs and myeloid cells. But this matrixellular protein is involved in cell-cell, cell matrix interactions. And we initially looked at thrombosponin 1 in a sterile LPS-induced injury model. And here, this is a little cartoon that we created. This is thrombosponin 1. It's a homotrimer. It's a large glycoprotein, and it's very sticky. And so many ligands bind thrombospondin 1, or actually many proteins bind thrombospondin 1 in vitro. But what it does in vivo is a little bit unclear. On its amino-terminal region, there contains a heparin binding domain. And this heparin binding domain is thought to interact with apoptotic moieties on senescent cells or dying neutrophils. And this enables thrombospondin 1 to bridge and enable recognition by macrophages of dying cells. Located within the TSR2 region is a sequence that can bind the clutch domain of a scavenger receptor called CD36. And it's this interaction that causes the triggering of IL-10, an anti-inflammatory cytokine, by macrophages during the resolution phase of LPS-induced injury. And that was what we sort of looked at in this particular project. Then we were very interested in different regions of thrombospondin 1, and one was in the carboxy region. It harbors a Cazal sequence, which is a sequence that confers serine protease inhibitory activity. And so we wanted to look at what this protein does in a live infection model with Klebsiella pneumonia. So we went on to show that this matrixellular protein curtails neutrophil granule serine protease activity that's extracellularly released during activation. So we wanted to study actually a bacteria that's very common in the ICU, Pseudomonas aeruginosa, behaves differently in the ICU than in chronic lung infection. And what's very interesting about Pseudomonas aeruginosa, and many of you probably study this, is that it employs very distinct virulence factors. Namely, it secretes exoproducts, including toxins, lipases, and proteases. And so in this proteolytic environment, what does thrombospondin 1 do? We knew that it actually inhibits the serine protease of neutrophils, but in this environment of proteolysis initiated by the pathogen, can thrombospondin 1 protect? And so the premise was TSP1 provides host protection against pathogen-initiated and host-sustained extracellular matrix proteolysis. In the thromboplastin 1 deficient mice, we show that there's impaired survival with PA14 infection. This is a hypervirulent form of Pseudomonas aeruginosa. It's a research strain originally identified in a burns patient, and we employed this to look at it mainly because there's actually a transposon mutant library, so we could kind of probe the pathogen side as well. And then here, despite when the knockout mice actually succumb, some of them actually do live, but the ones that do live show impaired resilience in the form of inability to regain weight after encountering the pathogen. So we felt like this was a good model to look at host resilience. To reduce survival bias, as you can see that the mice actually succumb by about day 2. We wanted to look at a time point before they die. And so this was about 20 hours. Here, what we show is that indeed the thromboplastin 1 knockout mice show impaired host events in the form of impaired bacterial burden in the lungs and splenic dissemination. These are all things I learned in Tom Martin's lab. And here, thromboplastin 1 knockout mice show enhanced neutrophilic inflammation. What we see is total BAL neutrophil accumulation in the knockout mice and total BAL protein, which is a surrogate marker of increased microvascular permeability as well as tissue damage. We also show here increased free neutrophil elastase activity in the air spaces, and then increased lung tissue pro-inflammatory cytokines such as IL-1 beta, IL-6, IL-17A, KC, which is a neutrophil chemoattractant, and lung GM-CSF. So these knockout mice show unbridled neutrophil activation and inflammation. Even though they have a lot of neutrophils in the lungs, they don't seem to clear the bacteria as well. So our question was, can TSP1 correct the hypersusceptible phenotype in the knockout mice following infection-induced lung injury? And so, as I said before, TSP1 is most abundant protein from alpha granules of platelets on a per mass basis. And you can see here, this is purified TSP1, this is platelet releasates from wild type, and these are from the knockout platelet releasates. And so we purified TSP1 from actually human platelets and gave it back to the mice. So this is the lung infection model where we have wild type mice, the thromboplastin 1 knockout mice, and then these are the thromboplastin 1 knockout mice given the platelet-derived TSP1. And you can see that there is mitigation of the impaired lung bacterial burden and reduction in cellularity in the air spaces, mostly comprised of neutrophils. And here's a good surrogate marker of total lung neutrophil content in tissue, and that's reduced, as well as lung any activity. So the question was, does TSP1 inhibit pathogen-encoded proteases secreted by Pseudomonas? So can a TSP1 mitigate some of this proteolysis that occurs with Pseudomonas aeruginosa infection? So I had a really talented resident at the time. She's now a fellow. She's now the Burns Wellcome Fund Scholar. And she actually screened a number of these Pseudomonas aeruginosa. And one, we started off with PA14. But PA14, if you take the cell-free supernatant after in vitro growth, you can see using a fluorogenocasian hydrolysis assay, you can see this is at 100% protease activity. And then Klebsiella pneumonia, which is a hypervirulent form, does not secrete proteases. But if you add aprotamine, which is a serine protease inhibitor, you can see a modest effect in inhibition of protease activity. E64 is a cysteine protease inhibitor, and then EDTA, which is a metalloprotease inhibitor. So based upon these experiments, we concluded that the majority of PA14 derived proteases that's secreted exhibits the profile of a metalloprotease. if you study and do a literature search on PA14 or any Pseudomonas aeruginosa, you can get a lot of hits. And we looked at the proteome of PA14 that was performed by other groups, and it turned out the most abundant protease, metallic protease, was LASB, which is a known virulence factor of Pseudomonas aeruginosa. And another name for Pseudomonas LASB is Pseudomonas elastase, because it can cleave elastin. So around the same time, we were looking at a number of papers, and we came across this interesting paper from Northern Ireland looking at the development of novel small molecule inhibitors, dipeptide inhibitor that targeted LASB with substantial in vitro activity against biofilm formation. And so we thought, well, maybe we could use this to look at it in our in vitro assay. So we generated this dipeptide inhibitor and screened the PA14 using the LASB inhibitor, and you can see this dose-dependent inhibition of proteolytic activity, as well as elastase activity, which I don't show. This is the EDTA as a comparator, and this is recombinant human TSP1 also as a comparator. So based upon this, at least in vitro, we could see inhibition of the extracellular protease activity secreted by Pseudomonas aeruginosa by not only thrombospondin 1, but also the LASB inhibitor. So our next question was, does disabling pathogen exoprotease activity correct the hypersusceptible phenotype in the knockout mice during infection-triggered injury? And so we used a transposon mutant PA14, LASB, and for a similar inoculum, you see here that the LASB mutant shows impaired, reduction in lung bacterial burden and reduction in BAL neutrophils and BAL protein as a marker of increased microvascular permeability and BAL-free NE activity. So disabling the LASB gene reduces bacterial burden, neutrophil activation, and lung injury in the knockout mice. If you disable the LASB using the LASB inhibitor following infection, so this is, I think, 2 hours post-infection, you can see that the knockout mice here show impaired lung bacterial burden, and then with the use of the LASB inhibitor, you can mitigate this response to normal levels. And this is BAL cellularity as well as BAL neutrophil accumulation. So if you knock out the type 2 secretion channel, so the protease LASB utilizes the type 2 secretion system to exit out of the bacteria to the extracellular space. So XCPQ is actually an important component of this type 2 secretion complex. And so if we use a deletion mutant of XCPQ, you can see here, this is the PA pathogen. We have the wild-type parent strain, PA14 here, and then this is the host, the knockout host. You can see impaired BAL, increased BAL protein, and then you can see a reduction in the BAL protein with the XCPQ mutant. And then here, this is free NE activity. You can see mitigation of neutrophil serine protease release, and then reduction in the MPO activity. And then here, then we ask, well, does it also inhibit NE activity as well? And sure enough, here, looking at human platelet release rates, you can get a reduction in percent NE activity. And then here, this is wild-type platelet release rate. You can see reduction, not as good as the human platelet release rate. And then this is the thrombospondin 1 knockout platelet release rates as a comparison. Purified TSP1 dose-dependently inhibited NE activity with human neutrophil elastase. And then this is human neutrophil elastase activity from neutrophils that we isolated from the tracheal aspirates of mechanically ventilated patients. We did microfluidics work with a colleague from MIT to get that going, and we see here reduction in NE activity with the addition of human platelet release rates in a dose-dependent manner. So in summary, we show that TSP1 provides host protection against pathogen-initiated and host-sustained extracellular matrix proteolysis. And now I wanted to then ask, well, if platelets are the major constituent of thromboplasmin 1, then does platelets have a role? And David Christiani had published this very interesting paper looking at a missense genetic variant. I actually never knew about this CARMEL1, and it actually improved ARDS survival. And he suggested that it improved ARDS survival by attenuating platelet count decline. So in other words, those folks who didn't have a decline in their platelets were protected by this variant, or was associated with improved survival. And now, I don't know if everyone knows, thrombocytopenia is an independent risk factor for ARDS mortality, and it's such a good marker for, predictive marker for sepsis. It's part of the SOFA definition. So platelet counts for all cause, low platelet counts are associated with worse survival. So we wanted to ask, what's the role of platelets in alveolar barrier repair following pathogen-triggered proteolytic injury? And so we were lucky enough to obtain the MPL knockout mice. These are knocked out for the thrombopoietin receptor. And so they actually have no observable defects just at baseline, actually do not die of a spontaneous bleeding diathesis. But what they do show is that they have a severe fatal injury with Pseudomonas aeruginosa infection. And you see BAL accumulation of hemoglobin. Most of this is membrane encased. It's not free hemoglobin. And you can measure this with OD540. It's a great surrogate for hemoglobin. And then if you look at the mice with Evans blue, it's a measure of microvascular lung leak. You can show higher leak in the MPL knockout mice. Interestingly enough, it's specific for Pseudomonas aeruginosa. We haven't tested other ones, but we did test a hypervirulent Klebsiella pneumonia strain, and it does not cause a hemorrhagic lung injury. If you look at the cell-free supernatant of Pseudomonas aeruginosa, so absent live bacteria, actually it's sufficient to cause lung injury in these thrombocytopenic mice. So this is PA14 supernatant. This is the vehicle. And then you can see increase in microvascular permeability as measured by BAL protein, and also increase in neutrophils in the BAL. If you ablate the PA exoprotease activity, you can attenuate the lung injury in these thrombocytopenic mice. And if you give wild-type platelets back to these mice, you can rescue the knockout mice from PA-triggered lung injury. Just to show that it's not simply maybe there's some fact or some other component of the MPL knockout mice that's causing this injurious response, we just depleted the platelets with anti-GP1B alpha. It's very common to do in the model system. And you can see that acute depletion of platelets using a monoclonal antibody targeting platelets, you can see increase in BAL protein, increase in OD540, as well as increase in BAL IgM, just another marker of microvascular permeability. So what happens when we dump down supernatant into the trachea of the MPL knockout mice? And we looked at these mice, and this is actually in collaboration with Center for Biologic Imaging at Pitt. And what you see is this is SPC marker in green. It's a great marker for type 2 epithelial cells. These are the progenitor cells that spread out to the alveolar spaces when the type 1 epithelial cells die. And you can see that there is cell death. And many of these are actually the ones in actually the pink is the DNA fragmentation that you see are seen with the type 2 epithelial cells. If you actually injure the lungs, and this is speculation on my part, the type 1 epithelial cells may die, but if you have enough type 2 epithelial cells to actually move into that space, you will eventually recover. But if you don't have enough type 2 progenitor cells, then you are in trouble. And this is probably what occurs, and this is my speculation, with acute exacerbation of IPF, which is a form of acute diffuse alveolar damage. We can model this using a lung epithelial cell line from mice, and this is live cell imaging with green as annexin 5, so you can see the progression of cell death. And if you boil the supernatant of the Pseudomonas aeruginosa to denature the protein, you can mitigate the cell death observed in the lung epithelial cells. If you use a pan-caspase inhibitor, the VVAD-FMK, you can mitigate cytotoxicity of the lung epithelial cells. But if you use necrostatin 1S, which is a necroptosis inhibitor, you fail to mitigate the cytotoxicity. And then finally, if you use XCPQ, the deletion mutant supernatant, you can mitigate the cytotoxicity observed. But if you use a triple knockout mutant of the type 3 secretion system, you fail to mitigate the cytotoxicity observed in this model. So based upon these experiments, we concluded in vitro that the supernatant causes lung epithelial cell death that is attenuated by the deletion of the type 2 secretion system outer membrane protein XCPQ. What it is probably a protein that's eliciting this response. And so this is just our in vivo, it's a very busy slide, but just the in vivo correlate of our in vitro studies where we looked at the XCPQ mutant, and for a similar inoculum given to the thrombocytopenic mice, what we show here is that the mutant reduces, you see reduced BAL hemoglobin, microvascular permeability, protein, IgM, and interestingly, the neutrophils are not different. And I don't show this data, but it's in the paper that we published that if you deplete the neutrophils, it actually does not mitigate the injury. Neutrophils are essential for fighting off Pseudomonas aeruginosa, as you know, clinically and also in mice. And here's the MPO activity. And this is a comparison with a triple knockout mutant of the type 3 secretion system, where you do not see differences in the OD540 or differences in the PA14. So we show that the supernatant causes lung epithelial cell death, and that's attenuated by platelet releasases. And here what we show is that for increasing the concentration of platelet releasases, you can reduce cleaved caspase 3 or caspase 3 activation, which is central to driving apoptosis. And so here what we show is increasing the concentration of platelet releasing. You can mitigate the cytotoxicity, and human platelet releasases had a much more dramatic effect. And this is just in vivo. We show that cleaved PARP, PARP is part of the DNA repair response, and caspases can cleave PARP. We can mitigate the cleavage of PARP with addition of platelet releasases in vivo, and then this is the BAL protein. We just use 100 micrograms of platelet releasases, and I suspect that if we did more, we could reduce it further. And so based upon this, we concluded that platelets protect against severe pulmonary complications from pathogen-secreted virulence factors that promote host cell death in the lung, even in the absence of clinically overt infection. I might add also that this thrombocytopenic mice was inspired by a patient that I saw when I was here at Fred Hutch as a junior attending. And a patient had diffuse alveolar hemorrhage and alveolar injury, and I think finally succumbed. But it was something that had always lingered in the back of my mind, is that even if you don't see infection, is it still possible to have alveolar injury with pathogen products? And so where are we going with this? Currently, we're developing a Pseudomonas aeruginosa database with ICUs across the UPMC centers, and we're prospectively collecting the first ICU Pseudomonas aeruginosa within respiratory isolates, and we're screening for proteolytic and elastolytic activity in vitro. Currently, we have about over 600 isolates in 500 patients. In the first 238, we do see that proteolytic activity and elastolytic activity is common, unlike chronic lung infections, let's say in cystic fibrosis patients. And there appears to be an association with 30-day mortality, even adjusted for Charlson comorbidity index. And we're looking at the high elastase producers and comparing them to the low elastase producers and confirming in vivo their injury response in mice. And then as a correlate to this, we generated a thrombospondin 1 flox mouse that by CRISPR-Cas9, and it sounded like a good idea. It took a long time, actually longer than I thought, but now we have the mice. We've actually crossed them with a platelet factor 4 cray, and actually it's a little bit premature to show, but they do exhibit a phenotype in this model system. And so if you haven't paid attention, the three things that I will leave with you today is that ARDS is relatively common worldwide, and two, it's still underdiagnosed and undertreated, and then three, infection is a common cause of ARDS and a common complication in ARDS patients. And so I just wanted to conclude with sort of going back to Dr. John Butler. And what I learned here at this institution as a fellow was really being enriched in a true community of colleagues. And so I would say that I've been privileged to collaborate with a number of colleagues, very diverse in their expertise, from me, but nevertheless, we've been able to foster some really intellectual projects together. And one is Prabir Ray, who's really mentored me when I was at University of Washington. And we looked at kind of IL-10 responses and non-resolving inflammation in a Klebsiella pneumonia infection model. And then we studied the mitochondrial damage-associated molecular pattern, cardiolipin, and how it blocks IL-10 production, causing persistent inflammation during bacterial pneumonia. And then another colleague of mine, Marion Kagan, is a lipid chemist, actually. He's really world-renowned for oxidative lipidomics. But actually, we were able to study mitochondrial and bacterial cardiolipin and how it modulates, this is extracellular cardiolipin released during a severe injury, how it modulates immunity, and it's an eaten signal for the phagocytic receptor that I discussed earlier, the scavenger receptor CD36. And then most recently, we've been looking at lipoxygenase. He's looked at it in the context of how this pathogen-derived lipoxygenase A, which by the way is a type 2 secretion system product, is turned on during biofilm formation and actually utilizes a host eukaryotic substrate, phosphatidylethanolamin, to trigger ferroptosis. And it's postulated that this is probably a way to derive nutrients from the host during biofilm formation, and this occurs during persistent lower respiratory tract infections. So I really want to thank you for your attention and giving me the opportunity to give this lecture, the John Butler Lecture. So thank you for your attention.
Thank you, Dr. Lee. Questions for Dr. Lee? Great.
Thank you, Janet, for that really interesting and stimulating talk. I have a couple of questions for you. So the first one is that you showed quite a bit of data demonstrating the effect of thrombospondin 1 on Pseudomonas-derived proteases. But obviously there are a lot of endogenously produced metalloproteinases implicated in ARDS pathogenesis. So how does platelet depletion or blocking thrombosponin 1 affect the response to other infections or causes of lung injury?
Oh, that's a great question. One is that we haven't tested other metalloproteases and other proteases that is secreted by the host, but I can tell you, at least in vitro, MMP2 and MMP9 is thought to be inhibited by thrombosponin 1. And then in other infections, I think others may have studied thrombosponin 1. And what I showed you earlier on with Klebsiella pneumonia was very interesting to us because it was kind of the opposite phenotype. And what it is that hypervirulent KP actually causes an early neutrophil response. And that neutrophil response is a phagocytic response, and that is critical for the containment of KP. So if you ablate that, actually what happens is that the knockout mice actually show a better phenotype. So I think it is a little bit context dependent. We haven't looked at other infections like gram-positive. We're trying to get to it. It's just that we have so many questions with the gram-negatives at this time. And in polymicrobial infection, I think it was protective, kind of similar to what we see with Klebsiella pneumonia.
So, Janet, you weaved a story around TSP1, platelets, Pseudomonas aeruginosa, and acute lung injury. Given the role or the involvement of Pseudomonas aeruginosa in CF, would you care to speculate about CF and its role in sort of lung pathology in CF?
Yeah.
At the same time, how about TSP1-based therapeutics for CF?
Right. Cystic fibrosis is very interesting because they have this kind of aberrant neutrophilic inflammation. And the thing about CF is that it's a chronic lung infection, and there's genetic adaptation of Pseudomonas aeruginosa. I don't know if Pradeep Singh is here, but that's very well studied, and they turn off LAS, like the regulator of LAS-B is turned off in many of the Pseudomonas isolates. Now, having said that, I think he did a wonderful paper with Peter Jorth when he was here, looking at different areas within the same lungs of the patient. And I think there may be some revision to that understanding. So what is actually interesting to us is that the type 2 secretion product, LAS-B, may be turned off. And then what turns on the lipoxygenase A, or does it concomitantly is it turned on? The LOX-A, which I showed you was critical for triggering ferroptosis, appears to only happen when biofilms are formed, so i.e. chronic infection. I think there is some movement to look at inhibiting endogenous proteases in chronic lung infections like cystic fibrosis, but I would submit to you if LAS-B is turned on, one approach would be just to target LAS-B because that seems to drive neutrophilic inflammation, at least in our acute lung infection model.
Janet, wonderful talk. Could you speculate on the host defense factors that you've outlined so elegantly in aging? So I would presume that as you get older, thrombospondin release is lower, but is that differentiated in different people?
Well, that's a fascinating question. I don't know if I have any experimental evidence that I've read or come across in the literature about that. That is actually very, very interesting. I do know that other cell types express and secrete thrombospondin 1, and I don't know if this is already published or not, but one of our colleagues was looking at scleroderma and skin lesions, and they did sort of a single cell RNA-seq, and it was presented in a conference, so I feel like I can say this. And basically what they showed was that one of the top candidates was thrombospondin 1. So it seems to be like an injury response. And there was a lovely Cell paper by folks at Harvard looking at thrombospondin 1 in the lungs in the context of bleomycin-induced lung injury and stem cells. And what they showed was that endothelial cell thrombospondin 1 is secreted, and it seems to be a molecular cue to drive progenitor stem cells to type 2 cell differentiation. Whether or not in aging, I'm getting back to the lung question, many of our interstitial lung disease are prevalent in the older population. And so perhaps there may be some regulation that's disrupted, but that's the only speculation on my part. That's a really good question. Okay.
I wonder if you've had a chance to look at the TSP1 knockouts later in the course of resolution, since TSP1 has also been implicated in causing fibrosis. I wonder if the TSP1 knockouts that do survive actually have less fibrotic resolution of their lung disease than
yeah, that's actually a very good point. So one of the binding partners and what TSP1 does is that it activates latent TGF-beta in vitro. And people in the original description by Jack Waller, when he developed the knockout mice, he showed spontaneous pneumonia. It turned out that's probably a dirty vivarium, and they are susceptible to injury for sure. But what he had suggested in that original paper was that because TSP1 activates latent TGF-beta in its absence, you can be susceptible to unmitigated inflammation and injury. However, I think there's been a revisit of that model, and myelofibrosis is one model where TGF-beta is an important molecule, and the knockouts do not have a significant phenotype. And in the bleomycin, it was actually a little bit of the opposite in some of the papers that I came across. So I think the thrombospondin 1 knockout mice, although it could activate the thrombospondin 1 in vivo could activate latent TGF-beta, there may be compensatory mechanisms in the knockout mice to overcome that injury. And in our model, we did it with the LPS-induced injury where we show the resolution phase, and for sure they have impaired resolution. We've been trying to do with the Pseudomonas aeruginosa infection, but the big problem is they succumb so early. So we've been really trying to down titrate the PA infection, but it's very difficult to get enough mice to look at that. But thank you for your question.
Any final questions for Dr. Lee? All right, let's give her one more round of applause. Thank you, Dr. Lee.